I'm a professor of cellular and molecular neuro-oncology based at the University of Portsmouth and run the Brain Tumour Research Centre there, which is core-funded by the charity simply known as Brain Tumour Research. I'm pleased to say that the Chief Executive and the Research Director of that organisation are here today supporting this venture. What makes Portsmouth a little bit different from other brain tumour centres is that we are apparently the largest lab-based centre in the UK, but also our concentration is exclusively on in vitro approaches, human modelling. We model the blood-brain barrier, and we use that to look at cancer metastasis. So we're investigating the mechanisms involved in circulating cancer cells adhering and penetrating the blood-brain barrier. That's all on the basis of human cell models in 3D. We also use that model to investigate drug therapy based largely around nanoparticle delivery systems. And we have other 3D models as well, like drug testing using normal non-neoplastic cells as well as cancer cells. And the idea really of using those human models is A, to get away from using animal models. And in my early career, there was pretty much nothing but animal models. So that was what we grew up with. But we realized that there were defects in those models and in a number of areas. Why we're using this sort of technology is to try to get agents fast-tracked into clinic. So this is particularly important when it comes to looking at the repurposed drugs. There are many drugs there which, when you compare them to the standard drugs which are being used to treat malignant brain tumours, as I do, malignant brain tumours are resistant to pretty much everything. Mean survival time for the most common and most frequent tumour is a mere 14 months. So these are a real challenge to overcome. But when we look at some of the repurposed and indeed the reformulated drugs, these stand up very strongly in comparison to the existing agents. Now the problem is it's difficult to publish our papers if we only use human in vitro models in the top journals, because they always want us to validate in an animal model. And I'm not sure validate is a useful word in this context. And the other thing is that trying to get money to fund this has been a struggle. I've been very, very successful, I would say, in persuading the Dr. Adwin Trust to part with money. It's very kind of them to support our work, not just recently, but over a number of years. And we thank them very much for their role in this particular development program. But I think it's important, if we're trying to fast-track drugs into clinic, these repurposed and reformulated agents, this may be a way forward where we're not going through hundreds of white mice and we're not delaying the process of drugs which actually we think have profound potential in treating patients. So how can we exploit this opportunity? I think one of the things that the brain tumour community and Sue Farrington-Smith on my right has been very much central to doing that is engaging the politicians in communication. We need to go behind the MPs and the parliamentarians, into the decision makers, into those people who are, what do we call them, civil servants, I suppose, who will help us to get the agenda much more firmly established. And I think we all need to work together in championing this in vitro, in silico, three-dimensional study work on human tissues, try and exploit this opportunity and work together. And we may come up with some nice ideas of perhaps how we can do that in a more collegiate fashion. That those people around this room obviously are signed up, I think, to that concept, and I'm probably preaching to the converted at this stage. But thank you for the opportunity of having a quick word. Be very happy to talk to other people about the systems we're using and to communicate with you about what you're doing and what your expectations are. But I think this is a fantastic opportunity, and congratulations to Kathy and to Jerry for putting this together to start the ball rolling.